Okay. So we shall begin with this webinar on biosafety considerations for gene-derived organisms. Greetings to all who have joined, all the participants. Good morning, good afternoon, and good evening, wherever you are. And I extend on behalf of International Society for Biosafety Research, a warm welcome to you all, and thank you for taking time out of your schedules and joining this interesting webinar on a very interesting topic. Just to give you a brief background in line with our objectives, the objectives of the ISBR to support the safe and sustainable use of biotechnology and facilitate the development of appropriate policies and regulations. ISBR has initiated this webinar series, a quarterly webinar on very important topics, and many of the suggestions coming from our ISBR members. And this is how we are now here. Just a minute, I don't know, it got stuck. So this webinar series is a topic-based webinars with the two presentations of 20 minutes each, followed by the discussion for 20 minutes. As I mentioned, we focus on the current topics which are relevant to ISBR members and continue to invite and request our members to suggest new topics. Two speakers will give different perspectives which will complement each other, and then we will have this discussion with questions from the participants. These webinars will be recorded and also published on the YouTube. We've had very successfully the first webinar where a large number of participants joined on data requirements, considerations for genome editing and new genomic technologies. This was held in March 2026, and today we have this second webinar, which is the biosafety considerations for gene drives. And then this will continue, and in the next year in 2027, we will have a symposium about which I'll explain you at the end. The background to this second webinar is that, as you are all aware, there are very active research programs underway using this novel technology, novel systems, gene-drive systems, with applications in animals, insects, and plants, most advanced in mosquitoes. However, translating this research to environmental implementation, to the use, requires robust, scientifically sound, and transparent biosafety frameworks. And a very positive initiative in this direction has been the adoption of voluntary guidance materials to support these LMOs containing engineered gene drives under the Cartagena Protocol on Biosafety. And hence, this topic is very important because this, continuing with this research now, various countries are also in the process of developing their guidance. We've had most recent document released by Australia for comments on the gene drives. And we have two very esteemed experts who will be talking today, Dr. Brinda and from FNIA Foundation for National Institute of Health, USA, and Dr. Felix Moronta Barrias from ICGB, Italy. So without taking too much of time, I would now like to invite first Dr. Brinda from FNIH, USA. Just to give a brief introduction, she is a senior technical expert and a gene drive policy lead at FNIH, and her work focuses primarily on biotechnology policy, global public health initiatives, and risk assessment and governance of engineered gene drive technologies. She has served as a technical expert and a policy lead at several international biodiversity and scientific panels, including the Convention on Biological Diversity, and was a very, very active member and instrumental in developing this guidance, which was adopted by the protocol, under the protocol in the last COP, and has a really very good experience in the methodologies which have been suggested in the guidance material. Over to you, Brinda, for your presentation, and I will just unshare this so that you can share your presentation.
Thank you, Dr. Viba and ISBR, Henny, and others for the opportunity to present on this topic. Let me share my slides. Hopefully you can see the slide deck.
Yes, we can see.
Okay, I will continue then.
Yes,
so
please.
Today I'm talking about the background and international deliberations that resulted in the development of this document, additional voluntary guidance materials, with a special focus on living modified mosquitoes that contain an engineered gene drive. This, as Dr. Viba mentioned, this was a specific issue of risk assessment that was identified in Cartagena Protocol COP-MOP decision CP 913. And the document that you see on your screen right now is called Biosafety Technical Series 7, Additional Voluntary Guidance Materials to support case-by-case risk assessments of living modified organisms containing engineered gene drives. So this is what I will describe a little bit about this document and how it was developed. So first, I want to say a little bit about what the Cartagena Protocol on Biosafety is, just in case there are people here who haven't heard about it before. This is an international agreement that obligates signatory countries or parties to protect biological diversity, both nature and ecosystems, as well as human health, from potential risks that could arise when organisms developed using modern biotechnology, what we call GMOs or LMOs, are introduced into the environment or moved across national borders. So a simple way to think about this is that the Cartagena Protocol provides the international rules of the road for ensuring that countries can benefit from modern biotechnology while also taking appropriate precautions to protect their biodiversity and human health. This protocol was adopted in 2000 and turned into force in 2003, and now has 173 participating countries that are parties. So this ensures that the parties have built or are building national biosafety frameworks and oversight capacity. Along these lines, the CBD has generated, with the help of ATEX and the parties, a practical guidance, this Biosafety Technical Series number 7. I refer to it as BTS 7 in short, just to make it easier to talk about it. But this is a practical guidance document that is developed to help countries assess the potential environmental risks of engineered gene drive, of LMOs containing engineered gene drives, particularly mosquitoes, with the use case of mosquitoes. Now, it's important to stress that this document does not create any new laws, any new frameworks, checklists, or requirements. This is totally voluntary and completely aligned with Annex 3 of Cartagena Protocol, in that it requires case-by-case comparative risk assessment. The one nice thing about it is that it introduced current best practices in risk assessment, such as problem formulation and pathways to harm, while still remaining aligned with Annex 3. And it provides guidance on what sorts of questions should be asked when evaluating safe testing or environmental release of these EGD LMOs, particularly LM mosquitoes, as the example. This document was welcomed by parties at COP-MOP 11 in 2024, and right now the document is available publicly, and the Secretariat for the CBD are encouraging the use of this guidance and the principles in it to do assessments. So with that in mind, I want to assure the listeners that BTS 7 was not created in a vacuum. In fact, I'm highlighting this process to show you that there was a long history of steps that were taken before we arrived at this document. So the topic was initially raised at COP-MOP 9 in 2018, where parties tasked the Secretariat for the CBD in their decision CP 913 to organize an online forum and an ATEG to determine if such a guidance was necessary for EGD LMOs. The recommendation from the ATEG in 2020 was to consider developing such a guidance, and this was taken up at the next COP, which is COP 10 in 2022. The decision from COP 10, which is CP 1010, set in motion the drafting process that resulted in BTS 7. In the intervening period, there were 2 online forums and a newly constituted ATEG that met twice to draft and then finalize this document in that intercession between 2023 and 2024. That draft document was, of course, taken to SBSTA in Nairobi in 2024, and then the parties discussed it at COP11 in Colombia and welcomed it at this COP in 2024, following which it was published as BTS7 and publicly launched by the Secretariat at the next SBSTA, which is SBSTA 27 in Panama in 2025. So just going back historically, in 2016, the Secretariat for the CBD published a general guidance on risk assessment of LMOs, which included mosquitoes as an example of the types and the types of traits of LMOs that should be considered, but it did not contain specific guidance on EGD LMOs, which is engineered gene drive containing LMOs. So there were also other guidance materials available at this time during this time frame that provided considerations relevant to EGD LMOs, such as the EFSA GMO panel, NASEM report. WHO has a report as well, and these focus on various aspects, including assessment, testing, and release, including challenges that may arise due to differences in the attributes of the EGD LMOs compared to other existing LMOs being assessed. So there are a lot of independent expert, national and international technical information and reports publicly available, and these fed into the ARTEC drafting group's deliberations. All of this work is reflected in the extensive reference list in BTS7. So if you look, there are more than 300 papers referenced in this report. And you might ask, why did gene drive containing LMOs require special attention? So as I mentioned, there are some attributes of gene drives that led to the decision that these type of LMOs might require additional attention from a risk assessment perspective under the Cartagena protocol. The first of these is biased inheritance. The driving nature of these sequences, the gene drive sequences, causes them to be inherited above the expected Mendelian frequency. We know this from nature because they do exist in nature, which means that their presence increases in shorter number of generations than traditional LMOs, so those without engineered gene drives. Beyond this characteristic, depending on the design of the EGD LMO, they may disperse beyond an initial release area and have a population level impact. And this may require considerations that are separate or different for risk assessment and risk management. And finally, certain use cases could also persist over long time frames and across many seasons, adding potential complexity to risk analysis. So BTS7 recognizes these features as key distinctions requiring additional risk assessment considerations, including what I mentioned: spread, persistence, removal or reversal or recall, potential for population level impact, and other genetic features that spread over large geographies and longer times, increasing uncertainty and creating potential challenges for risk assessment. So as I mentioned, the unique features of gene drive containing LMOs, particularly the issues of spread and dispersal over geographies, space, and time, were considered sufficiently different from LMOs regulated to date. And this also, along with the possibility of transboundary spread, required additional guidance. Along with that, there are uncertainties arising because this is a new technology from a lack of use cases, specificity and stability in the environmental interactions across large space and time, limited knowledge currently of behavior in the field, particularly over these long timeframes and spaces, effectiveness of reversal methods, and extrapolating from laboratory data and models as some of the reasons that motivated the development of this specific guidance with a focus on LM mosquitoes. So BTS7 is fundamentally about helping assessors address scale, persistence, and uncertainty, while it also provides a unique opportunity to use the foundation and framework of Annex 3, while including newer risk assessment methodologies such as problem formulation and pathways to harm. And like all other RTECs, this one was also a collective process. You can see the wide range of expertise from around the world, both parties, observers, and IPLC representatives working together with the CBD Secretariat's guidance, and as well the chair, who happened to be from Finland in this case, and with the input from comments received from the online forum and initial framework provided by ICGUB to draft the document. There were 5 drafting groups, and they worked really long hours during 2023 and early 2024 to generate the text, which was discussed during the second ATEC meeting and formalized into the document that was shared to Substa. So the document itself, the guidance, has a wealth of information across 7 sections. I show the sections here, and I will focus more on section 4, as this has the key elements of risk assessment, but there are 7 sections, 8 annexes on various topics, including modeling, uncertainty, and a glossary. And throughout the document, there are text boxes that highlight the mosquito-specific information to make that connection between the risk assessment methodology and the specific example of EGD-LM mosquitoes. The guidance also has numerous illustrative figures. Here is one of these, which helps to explain, the different types of features of gene drives that are being developed now and that could be envisaged for the use cases that would require assessment. Most of them are being currently developed for human and animal disease control and species conservation, with the primary driver being diseases that have a high death or disability burden, specific species loss, or economic losses, such as malaria, dengue, and currently New World screwball. There are 2 broad categories of gene drive applications being developed. One is for population suppression, where the aim is to extirpate the target population over time. The other is population modification, which is to spread a desirable trait rapidly through the population. And then they can also each be further subdivided based on presence in the population over space. So that would be spread and dispersal, or over time, which is persistence. And you can see that in the table shown in figure 3 from the document. Getting to the heart of the material, the most important information in terms of conduct of a risk assessment is outlined in section 4 of this document. On the left, you see the 4 practical questions underlying the rationale of the risk assessment process. What are we trying to protect? What could go wrong? How could harm occur? And then what evidence is needed, right? So this helps you to comprise what we call problem formulation, which is an early explicit step in the risk assessment that helps you derive from the national protection goals and legislation the problem that you are trying to solve, and thereby defining the scope and boundaries of the risk assessment, and then what the adverse outcomes are possibly are and how they should be investigated, thereby describing structure, process, methodology, and endpoints for analysis. And then finally, how these outcomes might be realized, thereby setting criteria for risk evaluation and what will constitute damage or injury. Throughout this process, there are opportunities to seek opinions of diverse stakeholders, including IPLC, and this is important to keep things transparent. Why this matters is that it helps regulators focus on the relevant questions, avoid collecting unnecessary data, make their assumptions explicit, and improve transparency and reproducibility. On the right of the slide, you will see how this problem formulation process aligns with the existing CP Cartagena Protocol Annex 3, particularly the steps in the risk assessment that are described in paragraph 8. So BTS-7, in summary, places problem formulation at the heart of the assessment process. If you are asking how does BTS-7 move through from protection goals to actual scientifically credible, measurable endpoints, it provides a bridge between these policy goals and scientific evidence by helping move the broad national protection policy objectives that are set naturally at a very high level to operational goals that are actionable and quantifiable. This table, again, taken from the document, helps explain how this is possible. So I will take an example of moving through the broader protection goal of biodiversity conservation and recasting it into an actionable goal, specifically in terms of ecosystem services provided by insectivorous bat population. So here we move from ecosystem services to bats and their role and the abundance of bats as a credible endpoint to assess. But this is only true if there is a credible pathway to harm linking EGD-LM mosquitoes to bat abundance. If so, then the operational goal would be maintaining bat population numbers within a given range based on pre-exposure baseline data, and related assessment endpoint for that would be bat abundance, with the endpoint capable of reliably reflecting the number of bats over time in the target area. The numbers changing beyond the range would require further scientific exploration to understand if this change was caused by the EGD LMO. So there has to be a causal link. So BTS7 will help translate policy objectives to risk assessment endpoints and then measurable scientific questions. And if you're wondering, I mentioned specifically scientifically credible pathway to harm connecting EGD LMOs to the bat population. This shows you a pathway to harm that is a sequence of events that must occur for the harm to be realized. In this case, the harm would be ecosystem services impacted because of bat population decrease. So the top, like I mentioned, the top level harm is ecosystem services, and the steps that have been outlined here test how the release of the EGD-LM mosquito could impact that bat population. And any of these steps in the pathway can have a risk hypothesis associated with it. The risk hypothesis would in turn determine the types of data that you would want to collect, which you see in the pale yellow boxes, the risk hypothesis in the blue box. The regulatory value of doing this sort of a pathway to harm is that any step that is unsupported by evidence that is collected or the data that is collected would reflect that this step is highly unlikely to occur and that the risk can be considered negligible, allowing the resources to be focused on other harms that are more likely to occur. So pathways to harm basically make the logic of the risk assessment explicit and open to scientific scrutiny and stakeholder input. So by nature, pathways to harm are ideally plausible scientifically, testable scientifically, and transparent and iterative. This is key. The value, as I mentioned, of the pathways to harm is transparency and ability to test scientific hypotheses. This is the basis of the scientific method in a nutshell. BTS7, the document, highlights this by recognizing the multiplicity of sources of evidence, including the use of models and tiered testing that can be used in a weight of evidence approach to inform, build, and test pathways in a precautionary manner, and acknowledging that uncertainty is something that is expected and characterized and managed as per the Rio Declaration Principle 15, which says this should not be a reason to postpone cost-effective measures to prevent environmental degradation. So the objective of this is to be able to use all the scientific information, stakeholder concerns, and input in a way that makes uncertainty visible, characterizable, and capable of decision support. And BTS7 highlights that monitoring is integral and a critical element of risk analysis. It devotes an entire section to monitoring because monitoring allows iteration of the risk assessment process, and it should definitely be not an afterthought. So monitoring highlights case-specific as well as general surveillance. The case-specific elements are direct outputs of the pathways to harm that I've discussed, such as what, where, how, and how long to monitor to ensure that risk hypotheses remain substantiated, as do the initial estimates of uncertainty. And the general surveillance description allows scanning for unexpected or unanticipated adverse effects that have not been encountered or addressed in the pathways to harm. So Together, this is a proactive way to continue to collect data from real-world use experience, whether that is in containment or in environmental use cases, to continue to test the risk hypotheses, validity of the pathways, and determine ongoing safety and efficacy of the product, which is the EGD-LM mosquito. Monitoring provides the evidence to validate or revise the original risk assessment. Note that risk assessment should be iterative, but the iteration has to be based on ongoing data that is collected from use cases. And in summary, I would like to leave you with 4 key points, and that is that this document is a voluntary tool that complements Annex 3. It does not replace it. It can be used for any LMO assessment. Problem formulation is the operational heart of the guidance. Mosquitoes are an exemplar use case. Pathways to harm and risk hypotheses improve transparency and scientific rigor and allow adaptive oversight. And monitoring supports iterative learning and is essential for managing uncertainty. BTS-7 is a practical framework for asking informed risk assessment questions. Its value lies not in providing predetermined answers, but in helping regulators and assessors identify the evidence needed to make scientifically defensible, transparent, and case-specific decisions. So with that, I will thank you and turn it back to Dr. Biba.
Thank you, Dr. Brinda, for a very nice presentation, giving the complete history, the contents, and the methodologies in the guidance, voluntary guidance developed under the protocol on a very, very important topic. Thank you so much. I request all the participants to hold on to their questions, put them in the chat box, and we'll have a discussion after we complete the second presentation. Now I would like to invite Felix for his presentation, and to briefly introduce Felix, he's a senior program specialist in the regulatory science group at the International Center for Genetic Engineering and Biotechnology, ICGEB in Italy. He specializes in biosafety, science policy, and biotechnology governance, assisting developing nations in regulating emerging technologies safely. He also leads the Monitoring Gene Drives initiative that is tracking gene drive developments for evidence-informed policy. Over to you, Felix. You can share your presentation and start. Thank you.
Yeah, thank you so much, Viva, for your introduction. And thank you so much also to the organizers of this ISBR webinar series. And also to you, Brinda, for that excellent overview. I think that your presentation provides a very useful framework for understanding how gene drive risk assessment can be approached in an structured and transparent way. So I would like to do what I would like to do in the next 20 minutes or so is to take a complementary perspective. Instead of talking about risk assessment itself, I will focus on the evolving evidence base that can help inform those discussions. In particular, I will draw on insights from Monitoring Gene Drive. That's a living literature review that I have been developing over the last year. So immediately now, I'm going to share my presentation.
Good. It's visible. Thank you. Go ahead.
Thank you for the confirmation, Viva. So, as I was saying, I'm going to take a complementary perspective of the previous talk. I will focus on the evolving evidence base itself, and in other words, what kind of technical developments, safeguards, and biosafety-related questions are increasingly appearing across the gene drive literature. And this presentation is based on a project that I repeat is called Monitoring Gene Drive, which is a living literature review developed here at the ICGEB. And just to avoid confusion, and this is important, In this context, monitoring does not refer to post-release environmental monitoring. Absolutely not. Here, monitoring refers to the continuous tracking of and synthesis of emerging scientific evidence. And why continuous evidence synthesis? Okay, gene drive is evolving very quickly. New papers appear across many different disciplines: molecular biology, ecology, population genetics, vector biology, social sciences even, and increasingly also biosafety and governance related discussions and so on. At the same time, the field is in itself is diversifying so much. So we are not longer discussing only malaria and mosquitoes. The literature now includes agricultural pests, invasive rodents, other insect species, even plants, and different types of engineered gene drive with very different characteristics and biosafety implications. And this creates a practical challenge. Even for people that work closely in the field, it becomes difficult to continuously follow the evidence base in a structured way. So, in traditional literature reviews, of course, we recognize that they are extremely valuable, but in a rapidly evolving field like gene drive, they can become outdated relatively quickly. So, how can we create a more continuous and accessible way to synthesizing emerging evidence that is relevant, in this case for gene drive research? So it is what is this project on monitoring gene drive. It was developed as a living literature review intended to continuously follow emerging research that is relevant to gene drive biosafety discussions. And the idea is not simply to collect papers, it's not a curation of paper, but to synthesize recurring questions and technical trends, emerging approaches, and also to consider uncertainties as the field evolves. And one motivation behind the project was the recognition that traditional review articles, as I said later, while they are extremely valuable, can become outdated very quickly, especially in fast-moving research areas. So the project was also designed to be openly accessible and easier to navigate across disciplines, including multilingual translations, newsletter, and shorter synthesis article. And one thing that became increasingly visible through this process of developing these literature reviews is that both the technology, but also the associated biosafety and governance discussions are evolving continuously and often simultaneously. In the rest of the presentation, I would like to briefly focus on three areas where the literature appears to be evolving particularly quickly. And these are the areas that monitoring gene drive is dealing with. First, the technical safeguard strategies and confinement approaches. Second, broader biosafety and risk assessment related discussions like modeling, for instance. And third, community engagement and participatory approaches that are increasingly associated with gene drive research. And of course, these three areas many times overlap substantially. For example, a technical design decision can influence biosafety consideration, and both can shape engagement discussion and so on. So I will begin with the safeguard strategies on how they are increasingly becoming a major area of innovation themselves. So a trend that becomes very visible when following the literature is the growing emphasis on safeguard strategies. Early discussions around gene drive often focused strongly on their ability to spread efficiently through populations. But over time, increasing attention has been given to questions such as how can spread be limited? Can system be geographically confined? can they be reversed or overwritten? Can persistence be reduced? Or how can uncertainties be managed during the phase testing? And as a result, the literature includes engineer approaches specifically intended to introduce different forms of confinement, controllability, or even reversibility. In the next slide, I will briefly highlight some of those examples. One important category includes systems designed to reduce the long-term persistence or uncontrolled spread. For instance, split drive system that separate key genetic elements into different genomic locations. And this means that the drive system tends to lose efficiency over generations because the components segregate independently. And a related concept is for instance, the daisy chain drive that were also proposed as a way to create a more localized or self-limiting spread dynamics. So to the audience, please mute yourselves because I'm hearing some background noises. Hello. Okay, I'm here. Okay, so I was saying that the daisy chain drive that it was also proposed as a way to create a more localized or self-limiting spread dynamics. And from a biosafety perspective, these approaches are interesting because they attempt to balance efficiency with a greater confinement at the same time. And for instance, how stable are these systems under realistic ecological conditions? We don't know. How predictable is their spatial behavior? Well, models can give us some ideas, but we really don't know. And one interesting aspect in the literature is that the same system can sometimes be presented simultaneously as a technical innovation, but at the same time a source of uncertainty. And with this, we can have an idea that how innovation in the design of the drive are expanding the toolkit for safer and more controllable gene drive. Another category is those that are dependent of the threshold and toxin antidote systems. Unlike highly invasive drive designed to spread from very low frequencies, these systems generally the threshold dependent system they generally require introduction above a certain threshold to establish their population. And this potentially creates greater geographical controllability and may reduce the likelihood of unintended spread following a small, let's say, hypothetical accidental release. And different architecture has been explored, including under dominant system and toxin antitoxin design. Well, the spread depends on crossing thresholds too, but the toxin limits the survival unless the organisms also carry an antidote. And from a biosafety perspective, I find particularly interesting that these approaches shift at least part of the discussion from simple spread efficiency toward a more ecological and population level dynamics. So, in other words, I would say that containment is no longer only a laboratory issue, but also a population genetic issue. And I wanted also to highlight other systems of safeguard that are reversal drive or immunizing drive or even the anti-CRISPR effectors. And the existence of these approaches does not necessarily mean that the reversibility is simple or warranted. Absolutely not. The thing is that there is an increasing attention to the possibility of corrective intervention if outcomes differ from the expectation. And this has important implications for biosafety discussions, of course, because reversibility is not only technical, but also affect the uncertainty, precaution, and of course, a potential phase testing while they are being conceptualized. I wanted also to highlight that the anti-CRISPR effectors field is very dynamic and it's growing so much. And these are examples of 4 articles that we have developed that are dealing with these strategies that I just commented. These 4 articles here. And now I'm changing the topic toward the ecological complexity and possible indirect effects of gene drive. So one recurring theme across the literature, and also we noticed from the previous presentation, is the increasing attention on to ecological complexity and possible indirect effects. And this is particularly visible in discussions around population suppression approaches. In many cases, the direct intended effect may be relatively clear, for instance, reducing or modifying a target population, but predicting the indirect ecological consequences can be much more difficult. So different studies have explored questions related to food webs, species interactions, competitive release, predator-prey dynamics, broader ecosystem processes. And one important point is that many effects are highly context dependent. So that means that same intervention may produce different ecological outcomes depending on the species, ecosystems, or even environmental conditions that are involved. So the discussion seems to me that are changing from is there ecological risk toward more under what ecological conditions might different effects emerge. Another important theme involves the challenge of ecological realism, and here modeling is something crucial. Many gene drive are initially studied under highly controlled laboratory conditions, but ecological systems, as we all know, are inherently variable and dynamic. As a result, the literature increasingly discusses the limitation of extrapolating from laboratory observation and very simplistic modeling approaches to more complex environmental settings. And this includes questions questions of modeling related to spatial dynamics, how patterns of space and connectivity influence spread and outcomes, and also the seasonality, how the temporal variation affects population, migration, the movement between population, genetic diversity, and also the uncertainty in parameters, assumptions, ecological processes that affect prediction and eventually decision making too. And talking about uncertainty, it is important that it becomes part of the discussion. And this is not because it is unique to gene drive. Absolutely not. Uncertainty is something that it is in any technology, in any human invention and human intervention. But the thing is that their potential scale and persistence of some system that may amplify the importance of long-term ecological considerations. And one thing that became very clear while synthesizing the literature is that gene drive should not be discussed as a single homogeneous category. And this is important. Different systems raise very different biosafety questions. For example, highly invasive suppression system may generate a different ecological concerns compared with localized approaches or threshold dependent approaches. And this has important implications for risk assessment, of course, because the evidence needed for or to evaluate one system may not be necessarily be appropriated for evaluating other. So the literature supports more case-specific and context-dependent approaches rather than generalized assumption about gene drive as a whole. And another interesting observation from continuously following the literature is that biosafety questions themselves evolve together with the technology. I already commented something like that, this part. I'm expanding a little bit here. As new safeguard strategy emerge, new assessment questions also emerge. As a system becomes more localized or controllable, discussion shifts toward different forms of uncertainty. And as research expands into additional organism or application, ecological and social contexts also diversify. So biosafety assessment or risk assessment are not static. They evolve together with the scientific landscape. And this is one of the reasons why continuous evidence synthesis can be valuable, not only to follow the new technologies, but also to follow how the surrounding biosafety discussions are changing over time. And increasingly, I repeat, this discussion also intersect with questions of participation, engagement, and decision-making processes. And with this mention, I transition to the last part about community engagement. But before, this is an example of all the other 4 articles that we have produced that deal with the ecological aspects of gene drive. Okay, and then finally we arrive to the engagement, community engagement. This is increasingly discussed alongside the technical development. This is the third major theme that this project is trying to address. And one thing that became visible is that engagement is no longer discussed only as a communication activity occurring after technical development. Instead, many publications now describe engagement, something that should accompany research and decision-making processes from early stage, even from the inception stage of a project. And this is especially relevant for technologies like gene drives, where proposed application may directly affect shared communities and shared environments. As a result, the literature increasingly discusses ideas such as co-development, stakeholder participation, transparency, trust building, and long-term dialogues. And these discussions are appearing not only in social science paper, but increasingly also in technically oriented publication. So this has been curious. And well, engagement is also a context dependent. This at the same time, the literature also makes clear that there is no one universal model for engagement. Different projects operate in very different ecological, cultural, institutional, and regulatory context. And as a result, many authors emphasize that engagement approaches need to be adapted to local realities rather than treated as standardized procedures. And another interesting trend is the growing interdisciplinarity of these discussions. Increasingly, publications involve collaboration across molecular biology, ecology, public health, ethics, social sciences, and governance and policy. And in many ways, this reflects a broader evolution in the field itself. And the questions surrounding gene drive are not only becoming more technically sophisticated, let's say, but also becoming more socially and institutionally interconnected. And as a result, the field is evolving toward more integrated, inclusive, and context-aware approaches. And in this category, for the moment, I just have one article already published. It's a huge article that honestly I need to update very soon. I would like to divide it in sub-articles because it's too dense now, but I'm proud of this article on community engagement on Gene Drive. So we arrived to the end of my presentation. So one thing that becomes very clear from continuously following the literature is that gene drive research is evolving simultaneously across multiple dimensions. The technologies themselves are evolving, safeguard strategies are evolving, and the associated biosafety and engagement discussion, of course, are following the same path. And this creates both opportunities and challenges for evidence synthesis and risk assessment, because the key questions are not only technical questions now, they are also ecological, contextual, and interdisciplinary questions. And for that reason, I think that continuously synthesizing the evidence base may help support more informed and adaptive discussion as the field continues to develop. And ultimately, one of the goals of this project of monitoring gene drive is simply to make these evolving discussions more accessible and easier to follow across disciplines and sectors. So finally, you have here the URL of monitoring gene drive, and here you have my email address in case of any further discussion. So thank you so much for your attention. I will be happy to discuss following questions.
Thank you, Felix. Thank you for very nice presentation, walking us through the developments underway and, this complex scenario. So one thing is clear, that all gene drives will require a case-by-case approach. And I think building on Brinda's presentation, problem formulation methodologies will be very useful. So I will now open it up with after these two very good presentations, I would open it up to the audience, and you can raise your hands. Before that, or put it in the chat box. We had a question from Professor Detlef that when and where do we expect first gene drive application in the environment? I know that translating this research and guidance to practice is going to be a big challenge, but maybe we would like to hear from our speakers on their views on, when do we expect? Brinda, you would like to speak first?
Yeah, so I see the question in the chat box, and the fact that in 2017 they predicted we would have something going to field in 2026. Now we are in 2026 and there's nothing there yet. It shows that it's not such a simple thing in the lab itself to develop a stable system that gives you the output that you want. That's not to say that they don't have some prototypes already. They do have some candidates, but it's not that straightforward for many reasons, not just regulatory reasons, even the science of it. There are some stressors that might help things that we hadn't anticipated moving faster, is like the case of New World Screwworm. There's not a lot of work being done in it, but there's a big pressure now with New World Screwworm moving through Mexico towards the rest of North America. This might progress the work in that space a little bit faster. There's always the case of rodents in Australia, and there is work happening on that. But the reason why it isn't moving faster is because mammals are hard to do the genetics part of it. And so science is trying, to keep up with the pressure that is coming from the field. So it's hard to predict which product will go to market first. Obviously, there's mosquito diseases, mosquito products that get most of the press. There is work in rodents. There is work happening in New World screwworm as well in North and South America. I don't know which candidate will go to market first or even to field testing first. So, Felix?
Yeah,
Felix, you would like to add?
yeah, I agree with your intervention, Brinda. Just to complement with, I mean, something that we have to keep in mind is that we need field tests. No, so we need to test. We would need to test. I mean, we as a humanity, no, as a human invention, and not because I am developer, but these organisms need to be tested in real condition. So before thinking in any application, public health application, they will need to get data from the environment. And this is only possible with field trials. So with small releases, perhaps you are asking that where developers are suggesting concrete island environment, oceanic island, or specific ecosystem that could perhaps provide some kind of geographical confinement, no, for the first field trials. So the essential step here, I think that apart from the data that are already available from related genetic biocontrol technologies like Wolbachia, sterile insect technique, etc., that are providing really good data of the effectiveness of population suppression and so on. I mean, the community needs data from actual gene-drive organism from the environment. So this is the key aspect. And as Brinda said, we were hoping like a decade ago for the first field trial to be happen within this year, something like that. So, and that's not possible yet. So it's difficult to predict, but let's see.
We can try. Let's see.
Yeah.
So we have one hand raised from Professor B.K. Tyagi. I'll take this liberty of extending the webinar time for a few minutes and give Dr. Tyagi, request Dr. Tyagi to give his comment. Professor Tyagi, you can unmute yourself, sir. Yes. Sir, you can unmute yourself. Professor Tyagi, if you can hear me, please unmute yourself.
Maybe he just asked a question for him.
Yeah.
Viva. He's having issues.
Yes. So you can type it in the chat box if you.
He did.
Okay, sorry. Oh, I didn't see that.
Yeah.
Dr. Tyagi has written, asked this question that starting from SIT 1960s, 70s through the little and now to the gene drives. Again, the same thing, how early we can make a breakthrough in mosquito-borne human disease like malaria or dengue. The journey seems longer than anticipated, something similar to which you pointed out. So there is an urgent need to deal with these problems for which gene drives have solutions like invasive species, mosquitoes, and so on. So any further comments from Brinda or Felix on Dr. Tyagi's question?
Not really anything related to gene drives, but yes, for new technologies to come to market, there's always a little bit of a lag. I mean, any technology, there's a lag to come to market because there's other things beyond science that need to happen, with the regulatory oversight, and those things can take time, stakeholder feedback, things that Felix has covered. So for any intervention to come to market, especially a new one that we've never seen before, it's always a little bit slower. Unfortunately, disease vector agents that spread these diseases, they are also mutating and changing and evolving. So by the time the technology comes to market, they've already moved ahead. So there's that as well to consider with tackling biological agents. And then there's something that neither of us mentioned, which is also like climate change and how it's impacting the spread of these vectors. And that is also happening in the same space and time as we are trying to get ahead of these things. So unfortunately, I don't have any good news for you, Professor Tyagi, today, but that's all I have to say on that. I don't know,
Yeah.
Felix.
And then this last
Yeah.
question, which is there. Yeah, sorry, Felix, you want to add something?
No, no. Let's go with the following question.
Okay. So there's
you.
one more question that, what further international guidance is useful or necessary for considering risk related to gene drive engineered organisms? I think you very nicely explained about the voluntary guidance material developed under the Cartagena Protocol. So do you think any further guidance is required?
For me personally, no.
Yeah, for you. Yeah, that's what
Yeah,
I
no,
mean.
I don't think that we need anything right now. And in addition to what we have, I think we just need to actually build capacity around what we have and
Yes,
get people to
exactly.
use it and report back on whether they think it's sufficient or not. But we need to put some dossiers together and we need to have regulators look at these dossiers.
So thank you very much.
Yes,
to
just
both-
quickly,
yeah,
Viva,
yes,
because
yes,
because
yes,
that
please.
that connect with
Yes,
my
please.
with my previous attempt to intervene, and that's very pertinent, Brinda. Thank you. And also, I would add that it's not only to use what we have now, that is more than enough, but also try to. I mean, because I think that it is a matter of what combination of scientific, regulatory, societal condition will be needed to enable a successful implementation or to enable a successful field trial. No, I insist in the field trial because this is the bottleneck now. So it's the combination of all these needed aspects.
Thank you. Thank you very much, Brinda and Felix, for the very great presentations on this complex topic. And thank you to ISBR for a board and the other people associated with it for, organizing this webinar. But before I close, I would like to share with the participants that those who are not aware maybe that ISBR organizes a symposium every 2 years, which brings together the global biosafety community to exchange knowledge, share experiences, and this is a great opportunity for collaborations and learning about the newer developments and so on. The most recent ISBR symposium was held in Ghent in last year, and now which had a participation of more than 350 participants from all the from various categories: scientists, regulators, policymakers, industry, communicators, and so on. So I invite you all to mark your calendars for the next ISBR symposium, which will be held in Bali, Indonesia in the October 2027. And also, I would like to also mention that the third webinar in this series will be on 26th October 2026. And the topic for the same is again a very interesting topic, AI designed protein sequences. So with this two information to all the participants, I thank all of you for participating and listening to these great presentations. Thank you very much. And I think with this, we will close this webinar. Thank you.
Thank you.
Thank you very much.
Thank you, Viva. Thank you,
Thank you.
Brinda. Bye, everyone.
Thank you. Bye.